myself, Dr. Khyati Maulik Kariya, at Mass General Hospital in Dr. Salvia Jain's lab, which is affiliated to Harvard University. This is my third year as a postdoctoral research fellow, post PhD. And today, thank you for this opportunity once again. And we are thrilled to discuss our recent finding about KLRG1 as a novel therapeutic target with you. But before I jump into the details, I must appreciate the backbone of our lab, Dr. Salvia Jain, whose awesome mentorship. It motivates us to continuously thrive to advance outcomes of patients with T-cell lymphomas. I would also thank here Bima, whose continuous efforts has driven the KLRG1 studies into action. As we all know, that despite the anthracycline-based chemotherapy regimens, with or without hematopoietic transplantation, and despite a plethora of new targets, the progression-free survival of patients with PTCL needs new treatment. One of the approaches is to treat malignancies with monoclonal antibodies to the surface molecule on these neoplastic cells to mediate effector cell functions such as antibody-dependent cellular cytotoxicity and antibody-dependent cellular phagocytosis. KLRG1 is one such target that is expressed predominantly on late differentiated TEM and TEMRA T cell population and NK cells. Accordingly, we expect its expression to be high in mature T cell and NK cell neoplasms. Might be expected, and a therapeutic antibody designed to deplete this KLRG1 positive cells might carry a potential cure for these disorders. And so here we define the molecular epidemiology of surface KLRG1 across various subtypes of mature T cell and NK cell neoplasms, and the potential for anti-KLRG1 monoclonal antibodies to deplete lymphoma cells from in vitro cell lines, in vivo xenografts, and ex vivo primary patient samples. So we started by examining the expression of KLRG1 across different TCL subtypes and observed a heterogeneous expression of KLRG1 at mRNA level and on the surface of the lymphoma cells in subtypes of mature T cell and NK cell lymphomas, as corroborated by our bulk RNA-seq analysis and protein levels from 64 patients' tissue microarray, which was extensively annotated and previously characterized in our lab. KLRG1 expression was observed to be heterogeneous in different subtypes of T-cell lymphoma, but we found that specific subsets such as ENK T-cell lymphoma or gamma delta T-cell lymphoma, TPLL and TGLL showed greater percentage of KLRG1 positive tumor cells than the other subtypes. We then sought to investigate whether this anti-KLRG1 antibodies could eliminate the leukemic cells from the plethora of peripheral blood of patients with KLRG1 positive T-cell lymphoma. We hypothesized that monoclonal antibodies that depletes the surface KLRG1 could be deployed for the eradication of KLRG1 positive lymphoma cells. The humanized afucosylated anti-KLRG1 monoclonal antibody, which we call it as monoclonal antibody 208, induced antibody-dependent cellular cytotoxicity and antibody-dependent cellular phagocytosis of TLGLL patient-derived CD8 positive, CD857 positive cells, X5O, and the subset of healthy donor-derived KLRG1 positive NK cells, CD4 positive, CD8 TEM, and TEMRA cells, while sparing KLRG1 naive cells and CD8 plus TCM cells, as well as B cells, thereby providing a potential therapeutic window of tolerability alongside activity. Next, we tested duvelisib, which induces apoptosis of T-cell lymphoma cells and repolarizes the macrophages to a pro-inflammatory phenotype, in combination with anti-KLRG1 monoclonal antibodies to enhance clearance of tumor cells. To gain further mechanistic insight into the transcriptional program and other proteomic modifications induced by duvelisib, we performed whole transcriptome sequencing of human monocyte-derived macrophages exposed to DMSO versus duvelisib at several time points. And overall, nearly 1,700 differentially expressed genes were identified between these two groups. And with notable increase in the expression of critical phagocytic factors such as phagocytic receptors, TLRs, CD14, as well as genes that are involved in the cargo degradation in phagolysosomes, inflammatory mediators like NALP6, and generation of reactive oxygen species. So subsequently, we treated a cohort of mice with duvelisib with the addition of anti-KLRG1 monoclonal antibody to investigate its reductive effects on tumor burden. Treatment of the cell lines as well as TCL patient-derived xenografts with monoclonal antibody 208 alone and in combination with the PI3 kinase inhibitor duvelisib extended the survival. Furthermore, the depletion of macrophages in vivo antagonized the monoclonal antibody to KLRG1 efficacy. Our findings suggest the potential benefit of a broader treatment strategy combining therapeutic antibodies with the PI3 kinase inhibitor for the treatment of patients with mature T cell and NK cell neoplasms. These findings have already been translated into a multi-center open-label phase 1 or 2 dose escalation trial, which Kusha can elaborate further.
Thank you so much, Khyati. I would like to start by highlighting the mission of our lab. We strive to improve the standard of care for patients suffering from a rare disease like T-cell lymphoma. The KLRG1 project, as flawlessly outlined by Khyati, emphasizes the advantages of the treatment combination of duvelisib, a commonly used kinase inhibitor that prevents cancer cells from multiplying, and the anti-KLRG1 antibody, a first-in-class antibody that can selectively deplete highly cytotoxic T cells while sparing important immune cells such as regulatory T cells and central memory T cells, along with many others. In humanized patient-derived xenograft models or PDX models of hepatosplenic T-cell lymphoma, HSTCL, and T-cell prolymphocytic leukemia, TPLL, this combination treatment truncated tumor burden substantially by leveraging the potency of repolarized macrophages. These findings, as Khyati mentioned, have been since translated to a multi-center phase 1 and 2 clinical trial for patients with relapsed and refractory T-cell large granular lymphocytic leukemia or TLGLL and is actively enrolling patients who suffer from anemia and or neutropenia. This trial aims to evaluate the safety, tolerability, and proof of concept of the anti-KLRG1 antibody in patients with TLGLL who are also anemic and or neutropenic. So far, 16 patients have been dosed in phase 1, and we are still dose escalating in cohort 5, which underlines safety, and we have seen promising improvements in blood counts for some patients. We hope that soon, anti-KLRG1 antibody provides a novel therapeutic option for many patients with TLGLL who show limited response to drugs like methotrexate and other non-specific immunosuppressants.